Hamadaweka, and then it's Daniel Hayden. Hello everyone, my name is Daniel Hayden. I'm a PhD student at the University of Wisconsin-Madison in the Department of Plant Pathology. Today I'm going to be talking the talk that I would have given in the actual conference that we would have had in the meeting over my work with microbes, specifically microbes in the soil and on the roots of plants, especially in maize systems. Before we get into like nitty-gritty science stuff, I like to do a land acknowledgement. I'm here at the University of Wisconsin. I mean, not like actually, I'm in my house, I'm in my apartment, but if I was, I'm in the same general area. University of Wisconsin-Madison occupies ancestral Ho-Chunk land, a place that their nation has called Job since time immemorial. In 1832 treaty, the Ho-Chunk were forced to cede this territory. That decades of ethnic cleansing followed when both federal and state governments repeatedly but unsuccessfully sought to forcibly remove the Ho-Chunk from Wisconsin. This history of colonization informs our shared future of collaboration and innovation. Today, UW-Madison respects the inherent sovereignty of the Ho-Chunk Nation along with the 11 other First Nations of Wisconsin. When I was able to say that, understanding, I think it's important, even as an Indigenous researcher at UW-Madison, to, try to further this idea of, making sure that all perspectives are heard in research. And so that's why I think it's really important to be working with like Native people, especially here in Wisconsin. So as I mentioned, I work in the Department of Plant Pathology, and what we do mainly is deal with the interactions between plants and microbes. So as we see plants interact with many different types of organisms on different levels, plant-plant competition, herbivores from insects and animals, and also pollination. Like I mentioned, there's also interactions with plants and microbes as well. We deal with what we know as pathogens, which are, the name plant pathology is we deal with like the pathogens that cause disease in plants, such as, it's like the Irish potato famine and other diseases like rust and weed. And also there are, it's not all microbes on plants are bad. There are good ones too. There, as we see right here, stuff like mycorrhizal fungi that are in the roots and help plants acquire waters and nutrients, and also bacteria that live inside the plants that can help with like drought resistance. So, but it's not just like black and white though. There is sort of like a middle ground where microbes are just living on the plant, because we know microbes are ubiquitous. They live on all different environments and different systems. So that's sort of what we're interested in my lab. So what we usually refer to that somewhat as like the microbiome, and so microbiome is just sort of referring to like the makeup of microbes in a certain setting. So a good example of that would be sort of when you hear people talk about like the gut microbiome. So in our gut we have lots of like bacteria and fungi in there, and so why we study that, or why people are looking at that, is because mainly the microbes are there, but they may play a role, in sort of, in this case, a role in like our health and other factors going on in our body. For example, they have fairly okay evidence suggesting that a certain ratio of bacteria, different families of bacteria, can be linked to certain obesity traits in people. That's just based on like the microbes living in your gut. So there's like lots of implications. That's why people are looking, and we do the same thing for plants also, because the main thing is plants living on the roots, because we know the roots are important for water uptake and nutrient uptake. And so there's a lot of work being done on sort of the microbes living on the root. And for our goal is to understand sort of what scientists are trying to look at the human gut, like what do the microbes do for the plant, like how do they affect the overall plant health. So and there, because it's sort of like an applicational side of it, where we want to, as if there's a way to alter the microbes living on the root or in the soil to perform a certain function, or to prevent disease, or prevent or allow plants to acquire more nutrients. There's different levels to it, how we haven't really gotten there, because we think about the ecology of microbes where they're all really small and there's a lot of them. We can't just count them, like how we count plants or animals. So we have to look at it from a different way. And of different types of ecology. This project also deals with some plant ecology also, and specifically with the factor of what we call like productivity, which is just sort of like how well plants do, or like that includes like how big they grow or how much seed they produce. And so this is a picture of my family's from. This is the Wichita Mountains in southwestern Oklahoma, and we see around here different plants. There's flowers, grasses, shrubs, and we know that these systems, like these grasslands, prairies, and forests, are actually what we refer to as the most productive, very productive systems. Meaning that with all like the diversity of plants there, all those plants living together do really well, and they grow really well together. And so a lot of people for a long time assumed, oh, okay, maybe it's just because these plants just work well together. Maybe, like, for example, the flower right here has these shallower, small roots compared to the grasses next to it, and they just don't compete for the same. Like, they compete in different areas of the soil, basically. Like, they're not competing directly. However, it's sort of, it doesn't explain too much, though, because we look, take birds or other animals, for example, that can eat different types of, insects or plants, you have animals that are pure herbivores, animals that are omnivores and carnivores. That's not really a thing for plants. Plants, they all need water, nutrients, and light. So, at some level, plants are always competing. And so, we're not really sure that the diversity in these systems are explained just by plants doing well together. And so, we're starting to look more at microbes in the soil and seeing how they actually affect plants, the diversity of plants in these systems. And so we look at stuff like resource cycling, where we know microbes are in the soil, they're cycling different minerals and nutrients that plants use. And so, for example, in legumes, nitrogen fixing bacteria and nodules. And then also, like, we know also that microbes living next to the roots produce a lot of, like, they break down a lot of molecules that the plants use to uptake. So that part of it can explain maybe just like, so like more plants means more environments for more microbes, and you have a large diversity. It's like, having a city with like different, with more housing, you just get more people coming in. But there's also another way science has been research has been looking at it, and that's what's known as pathogen suppression. And so what does that mean? How do pathogens play a role in maintaining this diversity and this productivity? On the left here is this famous plant ecology paper, and this graph's like really famous. It shows a basic ecological principle where the higher in diversity you are, the more productive you are. And it's sort of seeing this where they had these plots. One plot they had just one plant species, and then one plot they have like 30 plant species, or 32, I think. And then they measure that in plant biomass. Again, biomass is what you usually measure how productive a system is. So right here it shows that lower systems don't do as well, but the more diverse you get, the more productive you get. But at a certain point, it does just level off. And so that's interesting, meaning that we still see this trend happening. It's the more plants you have in a single setting, the more they'll grow. And so then this is sort of contrasted with this paper right here from more recently. And these black dots right here play the same role as these black dots, where species richness, number of species increases, the biomass also increases as well. And so these are all like field experiments done outside in like a natural setting. And so let's say you fumigate these plots. So let's say you have plots of like these different plant species, and you fumigate, meaning you eliminate a lot of the microbes in the soil. And there's a trend where you just sort of see like the line just sort of look like that, where it's just you don't really see a pattern at all. Which is interesting, because if you look at this one, lower speed, let's say when you get low right there, those lower species, their productivity is pretty low, and when you get higher, though, it's a stark difference in the soils that don't have microbes in it. So it's key right here to show that microbes do play a role, and we think specifically that it's just these pathogens. When you have low plant diversity, a low amount of plant species, you're sort of selecting for pathogens. If a pathogen that likes corn is in a cornfield, it's gonna have a great time. So if it's in like a large garden area, then it's not gonna have a good time, and it's gonna maybe get that those 2 or 3 corn plants growing in the garden. So this generally, when you bring in more higher diversity, you're bringing in more competition for pathogens. That's sort of the theory we think of now, and it's interesting because like these actually are controlling factors for stuff like why rare plants are rare, and why like you see more, and why invasive plants are able to take over environments a lot easier. And so for this project, we're sort of looking at how these different theories of ecology and for both microbial and plant systems, how we can sort of relate that to agriculture. And so we want to understand like how diversity and the above ground, the plants, affects that below ground diversity with the microbes on the roots. And then this will sort of help us understand, like how these may influence stuff like how big the plants get, how good they get at growing. And so in agriculture, that's what you want. You want your plants to grow big. You want them to do well. And so if we're looking at sort of high diverse systems agriculture, then we also want stuff that's like preserving soil health and weed suppression, which will be big parts of this project. And then for this project also, I'm aiming it more towards indigenous farming systems with native growers here in Wisconsin, because we know that native people have high diverse cropping systems. If we look at polycultures, this is like Three Sisters, or use of like cover crops, or even let's get crop rotation. We know that that's increasing diversity of what you're planting. And then also no-till or interseeding. We didn't have big equipment that could till up whole areas of soil. We usually hand planted and or minimum tillage, which your plant, if you do any sort of minimum tillage, your plant is still dealing with like weed pressure, everything else. So these systems are already diverse, and even further honing in on that, look at these varieties of crops. And in this case, for this project, we're looking at corn, and so we see all these different varieties of corn grown by different communities and tribes, and a lot of these haven't really been looked at in sort of a scientific lens. And it's an interesting point because oftentimes when people study corn in any sort of way, sort of making some sort of comparisons and contrast, when they usually compare those heirlooms or landrace varieties down in Mexico compared to just like some conventional hybrid, or some sweet corn hybrid, some modern one, they usually skip the large diversity that was formed in North America between domestication in Mexico, between conventional agriculture, and there's a lot there that can be looked at, and it could be an interesting lens to look at things. And then also we indigenous growing systems are more aimed towards what a lot of researchers are looking at for like sustainable agriculture and organic. So because the values of indigenous people for listing the land, having that cultural connection to land stewardship allows for these farming systems to be like this. And so for this first part of this project, it began the summer of 2019. It was basically just like a pilot experiment to see like what we wanted to do, what farmers were interested in. And so we started off working with the Oneida Agricultural Co-op, like the white corn group Ohelagu, and sort of what their main goal was, they grow white corn for both cultural and educational purposes. It teaches community members from planting to harvest, to even obviously in the picture right here, the like braiding and drying, sort of teaching and allowing community members to come together and learn these important things about culturally significant crop. And they're doing this sort of like on a larger scale as well. It's not just like a garden; it's like actual fields. So they're focused also on making sure they were doing things sustainably, and what the best way is, sort of combine these native corn varieties with some sort of conventional ag like mixture or methodology. So for this experiment, what we did, we had this small little like 3-acre field that we worked on, and we had these different treatments of interseeded cover crops. And so what that is, so you may have heard of cover crops, and oftentimes the most common use for them is whenever you cultivate a field during a growing season, farmers usually put in, it's a thing done conventional in like different types of agricultural settings, but you put in that cover crop after the growing season, because what it does, it's in the name, it maintains cover, so your soil is not getting too compact or too loose. You're not losing organic matter. Your erosion is not occurring. Any sort of leaching is not occurring. But then, and then also the idea is also you're increasing that plant diversity, which would in theory increase that microbial diversity, get that soil primed with microbes for the next growing season, who will then do those functions that I mentioned. This is nutrient cycling. And so, however, to reduce tillage, to reduce these methods of like interfering with soil, growers are looking more at like no-till and reduced tillage practices. And this one right here is just, interseeding the cover crops in between the corn, just planting right into it, is something that the white corn group was interested in looking at more. So that's what we did with these different cover crop combinations that were going in between the corn plants. So you have this really high diverse plant mixture during the growing season. And so how exactly do we study like, the microbes living on the roots? And so what I do is in our lab, I sample the roots off these different plants. This is like the roots of the corn, roots of this clover, and we take those roots and we extract DNA. We're not extracting corn DNA because we know it's corn. That's why we took the roots from corn, because we don't need to identify or do anything with the corn DNA. But we do want, so on each root, I see when you pick up a root in the soil, you see it's like a little dirt, but also living on that root is tons of bacteria and fungi. So we just break down the root and extract DNA from the bacteria, only from the bacteria and fungi, and then we sequence that fungal and bacterial DNA, and just to see what's living on the root exactly. And so then we run stats on that and just see how these communities living on the root differ. And this is sort of the sort of tough part about understanding this aspect of the project, because you think about it, you have let's say you have this root right here with different microbes living on it. And you have this root right here with different microbes living on it. There's thousands of microbes on these roots, and so and they could be completely different. So it's like, how do you compare that on like multiple levels? You have to compare root A and B. It's like root A, B, C, like root A with thousands of different combinations of numbers on it compared to root B with thousands of combinations of numbers. So it's like contrasting how those are different. You're doing that on multiple scales, multiple roots. And so how we do that is what's called this, how we sort of visualize that and do statistics on that is what's called a PCOA plot. And what you're doing here is you're sort of taking all those different, root communities with root microbial communities, comparing and contrasting them. It's not just X and Y, it's like X, Y, Z in different numbers of the alphabet. And you're seeing different like sort of dimensions, and you're sort of just like compressing that in a two-dimensional form. So we're sort of seeing the 2D dimension, like ideally a lot of these things would be sticking out the screen to feel like an actual going up the actual dimensions. But so the way you sort of read these, the closer the closer the symbols are, the more similar they are. So a good example right here is if you look at this pink one, number 6, clover and rye. So the corn plant, right, these two dots right here, and the cover crop plant, which is like a rye seed, or clover and a rye seed, rye root with these corn roots, they're close together, meaning that they're similar, which makes sense actually, because if you're they're all growing in the same space, these corn roots and these clover rye roots, so it makes sense they have similar microbial communities. And generally, it's, it looks really messy. These circles just work to indicate, like, it's just the code trying to draw these patterns for these different, treatments of cover crops, like with the communities, but they, it's just overlapping a lot. So that's sort of how you look at these, and we saw furthermore there was no significant difference in like the number of species or the diversity of species between each of these roots, meaning that it just all sort of overlapped. They were all somewhat similar, and so that was for fungi. This is for bacteria data. So it's something that helps sort of also help gauge and read these plots. Is right here is number 8. It says those 2 gray dots. Those are some blue corn samples from a different field nearby, and obviously it's like I said, if it's different, it's further away, it's more clustered further away. So this makes sense, why this is so far away from these right there. But again, we do see a constant overlap, meaning that there's no real difference in like the community compositions. They're all somewhat similar, and the reason why for that probably is just because we look at these treatments right here. There's not really any like sort of what we call factorial design. It's not just like, because like we have ideally in this factorial experiment you'd have if you had like A, B, and C, you'd have A, that's just treatment A, treatment B, treatment C, treatment A, B, a treatment A, C, and a treatment B, C. However, here we don't really see that, so it's kind of hard to gauge like what is affecting what, or what is the cause of like these patterns exactly. But it's just because we were just going at different combinations that would work to suppress weeds for the white corn group. And so with that in mind, we sort of had this idea for this next experiment to sort of try to hone in more of these differences in these. And so I worked with Dan Cornelius. He's an Oneida farmer, a technician for intertribal ag as well, and we had this field where it's very similar to the Oneida experiment, except we used different cover crops, and we were going to look specifically at productivity, how well did the corn do, then also the microbial composition, the microbes living on the soil and the roots, and also how this may affect the soil health further on. And for soil health factors, most differences in the soil take over like the course of several years, so we'd have to track this over time. So what we were working with here was we had that nice corn, the picture that you saw on the previous slide. It's a Oneida calico, and we had doing a similar thing with interseeding these cover crops in between the corn. And so right here are the different combinations. One is of wheat, and then next just clover, and then this one right here, for example, we have just wheat and clover, and then this chicory plantain mixture. So each of them sort of counts as like, we're sort of getting that kind of mentioned, that factorial design in where we're accounting for each plant growing in between. So if we know if a treatment does better, if a treatment with just wheat does bad, but a treatment with wheat and clover does really good, then that means clover is probably what's causing that. That's sort of how we're able to gauge like those differences through factorial design. And so over the course of the field season, things turned out pretty well. Right here's the corn growing. That's like I think a weekish or so after planting it. In these little green spots in between are the cover crops. And so yeah, there's more down there I think too. But yeah, so then here and also right here you see the same thing with the corn more mature, and definitely when the corn is getting big, you definitely see those cover crops. And then there it is further down the line. And like this is like late summer, and a big problem we ran into, because we're going to measure yield and look at how cover crops, these interseeded cover crops, these plants growing between the corn, affected how much the corn produced. However, we had a really big problem with deer and raccoons that came in and just would eat tons of corn. They literally ate, I would say, 80% of like the corn growing in this field, so we weren't really able to get a good gauge on yield. Here's just one of the cobs that I was able to nab from one of the raccoons, and it's really nice and it's really pretty, but it's just a shame that we couldn't measure that yield. But we still measure stuff like biomass and height of the corn, so we have some sort of idea of what yield could have been, or like how well the plants did overall. And so generally when we have these, as I mentioned, we have these different combinations of cover crops growing between the corn. These graphs are saying the same thing basically, but it's sort of looking at like the y-axis has the cover crop biomass, and then per plot, and this one has the weed biomass per plot, indicating like we had these, when we sampled, we took little squares in between the corn rows and just cut all the plants living there and separated them out, differentiating cover crop from weed. And so what this shows right here is that when you have more cover crops growing in between the corn, you have more cover crops to weed ratio. So meaning that, like, let's say 4 right here, that's pretty high. But then you have 4 right here, which has low weeds, and it makes sense because it's like around 6, one's around 4, so you get that 100% percentage. And so meaning that, okay, when you have higher species, they're able to suppress weeds better. When you just put in more cover crop, maybe they're competing more against the weeds when you plant them there. It's sort of like this idea of like a good neighbor and bad neighbor, where weeds are bad neighbors because they're coming in, they take a lot of nutrients, they grow wherever they want, they steal from the corn often. That's why people try to spray herbicides for weeds. But when you have a interseeded cover crop, though, it's sort of like what a good neighbor would be. They sort of grow there. They're not meant to like really take a lot from the corn, and they're also sort of meant to just like maintain a sort of suppression of those weeds. They're not allowing those weeds to come in. And so this is sort of taking those graphs and looking at them with a finer sort of look. And here's the different treatments at the bottom. And generally we saw this thing where these were probably the best at suppressing weeds, and all these treatments had this chicory and plantain mixture in them. And so these are what we call like these broadleaf forbs. And so for this experiment, we had the C4, the C3, like these right here indicate sort of like plant functional groups, as we call them. And when we look at different natural environments, there are different types of functional groups, meaning plants just do different things, whereas plants have like different like ways of going about themselves. And so for example, like C4 and C3 are just different levels of photosynthesis, but generally the C4 plant grasses are pretty robust and pretty big. And so corn is taking that role, for example, and C3 is taken by wheat, which is also a grass, as the C3 guys. And the clover and alfalfa in this case are like the legume, meaning that they fix nitrogen. The forb is like those leafy plants that you also see out in the natural system. So they're all sort of replicating natural systems in agriculture. And then also something we want to look at was so we know that those high combinations are better at suppressing weeds, but is it just because you put a bunch more plants in there that they suppress weeds, or is it because they actually were just good at doing their job? And so what this graph right here accounts for was obviously when we planted these higher combinations to agronomic standards, we did put in more seed. So the higher combinations, is number 7 right here, would have had more seed thrown in the beginning of the growing season. So that maybe explained why it did so well at suppressing weeds. There's so many plants there. However, what this graph shows right here is that sort of biomass to seed ratio. So what was the biomass payoff for the amount of seed you put in? And so a higher value would mean that's something that's good. That means you got more bang for your buck. And so number 3 right here, which is just nothing but corn and chicory and plantain, actually had significantly higher biomass seed ratio, meaning that you didn't have to put in like all these different combinations. You could just put in these 2 plants and you got solid biomass to seed. And we also see in the other graph as well, it was one of those ones that did pretty well suppressing weeds. So it's a fairly good trade-off to put these. So if you're thinking about using different types of cover crops, this would potentially be a way to go about it. Another factor we tried to take in is also like how did it affect the corn also. And so generally we didn't see any significant difference for corn biomass or height between any of the treatments, which is interesting because the idea behind these interseeded cover crops are, well, maybe they take away nutrients, they take away water from the plants, they compete with the plants, which to some extent is true, but in this experiment at least, we don't really see that. And it could also be the fact that these indigenous corn varieties may be a little hardier. They may be used to historically growing with other plants as well. That's sort of a theory that I'm spitballing around a little bit, but it's just like this idea that maybe for these indigenous corn, intercropping cover crops could actually be a good thing. And so right here is we're seeing just this general somewhat small relationship between biomass and height, meaning that when you increase in height, the biomass of the corn did increase, but generally it's like not too strong, I would say. And I mean, and even further, this can sort of be explained also. What we have here are the blocks. So this experiment, you can think about what block means, like there's different replicates of the experiment. So we grew those 10 treatments 4 different times. And in block 1 and 2, we actually the corn plants were a little taller in general. As you can sort of see, there's more black and red dots to the right side of the screen. And that could be attributed to several things. One could have been when we tilled up this part of the field initially, there could have been more initial nutrients available on that side of the field for the corn. So for example, how we sort of set the experiment, it was like a long rectangle field in block 1, block 2, block 3, block 4. So there could have been more nutrients right there that helped the plants get a little taller, or it could have been something with the weeds. We aren't really sure, but it didn't seem to affect biomass though, because biomass is still significant between different treatments. And so that's sort of it where we're at right now. I'm still working on a few more data with the cover crops, and then I'm also processing a lot of the root samples in the lab right now to be sent off for sequencing. And so for this coming next field season, I'm still continuing my work with the Halagoo. I had to essentially cancel field work for the season up in Oneida just because of how bad the COVID-19 pandemic hit that county really hard, so I just didn't want to really risk anything. And but next year though, we're continuing that. We're going to continue more on focusing on what agronomic methods can be better done for weed suppression, because I know they have a big problem with weeds this year. And then also I'm going to do a greenhouse experiment. And so what we can do in the greenhouse is, as I mentioned that one experiment earlier on where they fumigated the soil, we can do that, but more precisely, we can just sterilize the soil and grow it in the greenhouse, so we can have those corns growing with those different cover crop mixtures. And what sterile soil can do to these kinds of experiments, so let's say we see a phenomenon between plant A and plant B. We see right here plant B is a lot bigger than plant A, so we have a pot with plant A and plant B. And so if we sterilize the soil, so we see it's like regular soil, plant A is smaller than plant B. If we sterilize it in plant B, it is the same size as plant A, then we know that by getting rid of those microbes, we prevent that they were playing a role in making sure that plant B was taller. But if we sterilize the soil again, or we sterilize the soil in a different scenario, and we see that plant A is still smaller than plant B, then we know it's just the plants, basically. Maybe plant B is just really good at competing with plant A. So that's what we're able to do. We're sort of able to narrow down, is it the plants doing the work, or is it the microbes doing the work? Or it could be both to an extent. Then next, I'm working with Dr. Frank at kind of Menominee Nation College. We're going to run some corn experiments and to see what microbes are growing on these different corn varieties in the roots. That's something that I was unable to do this year, but want to do, this fall and next spring, is get more native growers involved in this project, especially ones that are doing a little more like higher, larger scale experiments where they would need stuff like interested in cover crops would be interested in doing that. And so I'd be happy to work with them as to expand the project, expand UW-Madison's research to make sure that perspectives of indigenous growers are taken into account when we sort of do our research and our science. So with that, I'd like to acknowledge my advisors, Dr. Aaron Silva and Dr. Rick Lankow. Aaron Silva, she's the organic extension agent for UW, so she's played a pretty big role in understanding the sustainable agronomic side of the project. And Rick Lankow, he's a soil ecologist, so he works a lot with like the soil microbes. So that's sort of two sides of the coin right there. And then I'd also like to acknowledge my Oneida collaborators, Laura Manth and Tony Kuchman, all of the White Corn Group, Ohelagu, and then Dan Cornelius also for allowing us to work with him for this field season, and he's continuing. We're going to continue that same experiment for next year also, and so he's really interested in sort of what we got going on and taking like the advice and the research for next year. And then I'd like to thank my labs also for providing assistance for the field season. We're real short-handed because of COVID, so people coming to help was nice. And then Department of Plant Pathology, UW-Madison. My funding sources are the Ford Foundation and the Center for Integrated Agricultural Systems at UW-Madison. And yeah, here's my email up here if y'all have any questions. Of course, if I was at the actual meeting we're going to have, I would ask if anyone has any questions and answer them there, but we can talk more. But please feel free to send me an email. I'm always happy to talk about these things. I'm always happy to collaborate. Ideas for collaboration also. This is Dan Cornelius' field right here, and this is where we're interseeding straight into the ground these corn, and they're not too shabby actually. I mean, my advisor said, she does a lot of no-till experiments. She said these look pretty good for no-till. I think also it's just thinking about, a lot of the knowledge that's held within these, corn that we don't really know yet, or we don't know too much about, and thinking this is like the picture what we want. This is what we want to see. Minimum input for agricultural systems, but yet at the same time preserving and maintaining healthy soil and healthy plants that can be used to make healthy food for healthy people. So sort of creating that pipeline from microbes in the soil to us and eating this food. And so like I said, if you have any questions, feel free to email me. And thank you very much. Hope you all have a good day whenever you watch this video, and y'all stay safe.